Hello and welcome to our presentation of ethanol production from yam pills using Aspergillus niger and Saccharomyces cerevisiae. Here's the list of the content, which is introduction, project description, research and design method, budget and cost analysis, entrepreneurial opportunities, and conclusion. Let's get started!
A sustainable way to deal with agricultural waste and produce renewable energy is to turn yam pills into ethanol. Due to their high carbohydrate content, yam pills a plentiful residue of yam consumption and processing, which makes an excellent feedstock for the synthesis of ethanol. The hydrolysis, fermentation, and distillation procedures used to turn yam pills into ethanol are the main topic of this study. While discussing possible difficulties in large-scale production, the paper also emphasizes the financial and environmental advantages of using yam pills as a raw material. For our project, we are using Aspergillus niger and Saccharomyces cerevisiae to hydrolyze and ferment yam pills for the purpose of producing ethanol. In addition to tackling environmental issues like waste management and pollution, this study emphasizes the use of agricultural waste as a renewable resource that helps create sustainable energy solution. Pills from yams, which are frequently thrown away as waste, are a good source of carbohydrates and can be used to produce ethanol. By maximizing variables like fermentation duration and ethanol concentration, the study examines how well these microbes can convert the yam pills into reducing sugar and then ferment them into ethanol. The project's goal is to create an economical, green bioethanol manufacturing process that decreases reliance on fossil fuels, lower greenhouse gas emissions, and promote economic growth. These projects provide the combined advantages of solving environmental issues and developing a sustainable energy source by utilizing agro-waste, especially in areas like Ghana where these resources are easily accessible. The tissue culture technique used in this project involves isolating and cultivating microorganisms for ethanol production. To isolate Aspergillus niger, yam pills were chopped into small pieces, sterilized with 1% of sodium hypochlorite, and placed onto potato dextrose agar (PDA) plates using sterile forceps, where the growth was observed within 48 hours. Pure culture were obtained through subculturing. For the cultivation of Saccharomyces cerevisiae, a strain was obtained from Fleischmann dry active yeast, diluted and inoculated onto PDA plates. There is a potential commercial benefits from this project. This initiative offers significant economic advantages by converting abundant agro-waste, such as yam pills, into bioethanol, providing a sustainable and cost-effective alternative to fossil fuels. It can also reduce fuel import costs and enhance energy security, particularly in regions like Ghana. The production of bioethanol fosters agro-industrial growth by creating a value-added products, supporting local economics, and addressing environmental issues by reducing pollution and greenhouse gas emissions from traditional waste disposal. On the other hand, it can also generate jobs in waste collection, processing, and ethanol production while benefiting farmers through increased demand for crops and their byproducts.
For our project, we will be converting carbohydrate-rich yam pills and agricultural byproduct into bioethanol using Aspergillus niger for hydrolysis and Saccharomyces cerevisiae for fermentation. The process overview, we will use hydrolysis, in which we will hydrolyzed yam peels, our yam peels will be pre-treated and broken down into simple sugars by Aspergillus niger. And we will also use distillation in which ethanol is purified for commercial applications. Finally, we will use fermentation. The sugars are fermented by Saccharomyces cerevisiae to produce ethanol. The process in which we produce ethanol from yam peels will take about 5 weeks, in which the first 2 weeks we will focus on sample collection, drying and preparation of the yam peels. In week 3, we will undergo hydrolysis and daily analysis. On week 4, we will undergo fermentation and daily monitoring. And finally, on week 5 is the distillation and final ethanol analysis. The outcome of the process is that ethanol yields up to 31.86%. This is eco-friendly approach, reduces waste, supports renewable energy goals, and provides a sustainable alternative to fossil fuels, promoting environmental and economic benefits.
This is the research and design method. The outline of the procedure started with sample collection and preparation. Plantain and yam peels were prepared for analysis by cleaning, peeling, drying, and grinding into a fine powder. The second step is laboratory analysis. The first analysis is Aspergillus niger. Yam peels were cut, surface sterilized with 1% sodium hypochlorite, and then cultured on potato dextrose agar (PDA). A pure A Niger culture was obtained through subculturing. The second is Saccharomyces cerevisiae. A pure Saccharomyces cerevisiae culture was obtained using cereal dilution and multiple incubations on PDA and yeast extract agar. Preparation of culture media for the hydrolysis of yam peels part. A 50-gram mixture of plantain and yam peel flour was sterilized and inoculated with Aspergillus niger. After 7 days of incubation with daily shaking and sugar measurements, the cultures were filtered. Next is estimation of reducing sugar. The Nielsen-Somogy method, a colorimetric assay using DNSA, measured reducing sugar concentrations. Absorbance reading calibrated against a standard curve provided quantitative data on reducing sugar levels in the samples. Fermentation. Yam and plantain peel filtrates were fermented using Saccharomyces cerevisiae, with some cultures also including Aspergillus niger, followed by sterilization and incubation. After fermentation, the mixtures were filtered and sterilized again before distillation. Here we go for the last part of preparation of culture media, which is distillation. Ethanol was separated from the fermented broth by distillation at 78 degrees Celsius and quantified using an alcohol meter. This experiment assessed bioethanol production from yam and plantain peels using different microbial combinations, comparing their ethanol yields.
Yeast nutrient. Yeast nutrients play a crucial role in promoting the growth and activity of yeast during fermentation. And fermentation is the key for converting raw materials like sugar into ethanol or other byproduct, which is a core part of this project. The cost varies from Rm2 to Rm8 per kilograms. It's depending on the type and quality, and higher N nutrients result in more efficient fermentation. And sugar source. Sugars are the primary food source for microorganisms, especially yeast during fermentation. In ethanol production, yeast converts the sugars into ethanol. The cost of sugar source range from Rm 80 cent to 3.20 ringgit per kilogram, depending on the type of sugars used, such as cane sugar or glucose syrup. More refined sugars tend to be on the higher end of price range. Growth curve. Growth curve consists of microorganisms like Aspergillus niger or Saccharomyces cerevisiae yeast, which break down complex sugars and produce ethanol. The cost of cultivating these microorganisms varies from Rm20 and Rm80, depending on the factors like this type of microorganisms, production, sales and cultivating materials. Yam peel, the raw material. Yam peel is used as a raw material providing a carbohydrate source that can be broken down into fermentable sugars. It's often selected for being a waste product from agricultural processes, making it both cost effective and sustainable. The cost of yam peel range from Rm 40 cent to Rm 2 ringgit per kilogram, with variations based on availability and season.
Next, I'll be sharing insights on business opportunities, potential partnerships, the business model, and expected income for this innovative venture. Let's start by exploring the many ways to grow this business. To expand the industry, we can build a strong supply chain with collecting yam peels and other agriculture waste from farms, markets, and food processing facilities. This ensures a consistent supply of raw materials for ethanol production. Expanding product offerings to include ethanol-based items like sanitizers and industrial cleaning agents open up opportunities in healthcare and manufacturing. Additionally, providing waste management services for food processors help reduce environmental impact and lowers disposal costs. Training sessions and consulting services can also support new entrepreneurs entering the ethanol market. Forming partnership with government agencies and environmental organizations can further enhance operations and promote sustainable practices. There are excellent collaboration opportunities with research centers in Malaysia. For example, the Centre for Biofuel and Biochemical Research (CBBR) at University Technology Petronas developed ways to turn waste into ethanol and other biofuels. Another promising partner is the Biotechnology Research Centre at University of Nottingham, Malaysia, which focuses on sustainable agriculture and biotechnology research. Revenue prediction shows strong potential for growth. In year 1, producing 1,000 liters of ethanol per month at 10 ringgit 35 cents per liter would generate 124,200 ringgit annually, with an additional 2,250 ringgit from byproduct sales. By year 3, production could scale to 5,000 liters of ethanol per month, generating 621,000 ringgit annually. 11,250 ringgit obtained from byproducts will be added with total revenue of 632,250 ringgit. This highlights the profitability of turning agriculture waste into biofuel while promoting sustainable development.
And coming to the conclusion, this study highlights the effective use of yam peels as a sustainable feedstocks for bioethanol productions using Aspergillus niger and Saccharomyces cerevisiae. This process not only addresses waste management challenges but also promotes renewable energy solutions, reducing reliance on fossil fuels and environmental impacts. This innovative approach demonstrates the potential for agro-waste utilizations in creating sustainable and economically beneficial biofuels. Thank you.